these primary and secondary, all this needed ATP to operate, and then you build up the concentration gradient, and then you can use this, right? The other type of one that also uses energy, right? And we don't really talk about how it uses energy, but the formation of vesicles, right? To bring in stuff in bigger chunks inside the cell or release bigger chunks outside the cell, right? This will be vesicle transport, right? All these vesicles either bringing stuff in or out, endocytosis and exocytosis. So while we're talking about that, we'll bring up a topical subject, right? This is a virus, and they are specialized at kind of binding to certain receptors, right? If this was the HIV virus, this would be a certain receptor. As if this was a coronavirus, this would be another receptor. But they, these receptors are like, locks, and the viruses have a key. So once they bind to those, they have this whole endocytosis action right here, which brings the cell, which was having these receptors for other purposes, has been hijacked. And now the viruses get inside the cell, and then they do their stuff. They kind of capture the cellular making machinery, make more copies of themselves and all their parts. And then they are released out into the environment, right? Or do whatever they do, right? But so what the virus is doing in this case is exploiting these sort of natural endo and exocytosis activities right here. So this is all to say all this whole other crossing the cell membrane stuff, transport in vesicles. Your vesicles, this picture kind of shows it a little more clearly. You got the lipid bilayer. In this case, this is an endocytosis, right? You get this kind of dip made out of the cell membrane. And so these vesicles are formed from the cell membrane. They're the same kind of lipid bilayer structure, except they're just a little circle with a couple of little things in it, right? And so this will go for endo or exocytosis. You're either making it, pinching off the cell membrane, or you're going to be merging with the cell membrane and releasing its contents, right? Endo and exocytosis. And again, this is somewhere along the line it's an energy dependent process, right? ATP is going to be needed to form all these vesicles and fuse with the membrane and stuff. So for these, we're going to have endo and exocytosis. Within endocytosis, there's 3 different types or categories of types of endomitosis, that we'll discuss very briefly here, right? You're going to have this phagocytosis, cell eating, penocytosis, cell drinking, and then this other receptor-mediated endocytosis like we talked about with that virus, right? So here is one of these macrophages. Over here, this is one of your immune type cells. It goes around your loose connective tissue and finds things like dead cells or damaged cells or pathogens like this bacteria here. And it's basically kind of has this amoeboid motion where it extends, its parts of its cell body right here and basically eats whatever it is, like takes it in and then busts it down and digests it within its own cell right here, right? So this is phagocytosis, otherwise known as this cell eating over here. Right? Digesting these large particles right here, bringing them into the vesicles, and then it fuses with these so-called lysosomes. And they're going to break it up, and then they can release the broken down particles right here. Right, so this is your endocytosis. Macrophages are a big one. These are the macrophages, and they eat stuff for different reasons, right? Either because they're a pathogen, or maybe it's a dead cell or stuff like that. All right, so that's phagocytosis, cell eating, so-called. And then penocytosis is referred to as cell drinking, because again, you're just going to have the plasma membrane folding inward. It's going to form a vesicle, and whatever is around here is going to be brought into the cell like this, right? So this will be the extracellular fluid and whatever dissolved solutes are in it, right? They're going to be brought into the cell in this fashion, rather than through the lipid bilayer, through channels, right? You're just basically taking these small molecules, small particles, whatever is in the interstitial fluid, into the cell right here, right? And, you know, if there were stuff you needed for digestion, you can fuse with these lysosomes, right, with the digestive enzymes, and use whatever was in here for whatever you're going to use those for. Right? So this is happening in most of your body cells. They're just bringing in whatever was in the extracellular fluid for use of whatever purpose. So this is just happening. There's no so-called receptor proteins involved, unlike our receptor-mediated endocytosis. Okay, and this one, just like, you know, the viruses do, in that case, there is a receptor called the ACE2 receptor that it binds to, and then those ACE2 receptors are going to be associated with these clathrin molecules, right? So you have this clathrin-coated pit, right, that kind of causes this invaginated clathrin-coated plasma membrane, kind of forms this vesicle over here, and then the cell does whatever it might do with them, right? So this is where your cells take up cholesterol, right? Remember those vesicle, those lipoproteins that were floating around, right? So these LDL particles are going to bind to these LDL receptors and then be taken in by the cell, right? So it's not just cell drinking or cell eating, it's very specific to these receptors, right? The molecule, whatever, has got to bind to these receptors, and then it's taken into the cell right here. All right, so this is much more particular than cell eating or cell drinking. It has to bind to these receptors first before it's brought in. All right, so that's, again, you know, for our virus purposes, right? This is what is happening. In this case of the coronavirus, there's this ACE2 receptor that it recognizes. It just specializes. It's found a way to kind of connect with that and cause this invagination event, and then it's brought into the cell, and then it does its thing, right? So that is receptor-mediated endocytosis. All right, so that's bringing stuff in, and we also sometimes want to release stuff out of the cell, right? So in this case, you're forming vesicles, usually in, you know, your Golgi apparatus or something like that. You're packaging a bunch of proteins or whatever you want in here, and then those vesicles are going to go and fuse with the plasma membranes, and then the contents are going to be released into the surrounding space right there. Right? And this could be a regulated one, right? Maybe, as we'll see with, like, say, calcium triggered release, as we'll see in the nervous system, or it just could happen normally, right? And we're going to see this in these secretory cells, which are going to secrete digestive enzymes, hormones, mucus and stuff. Remember, those epithelial linings and glands that secreted stuff. They're all going to do it through some form of exocytosis, right? And then we'll also see it in the nervous system, because the nerve cells are going to release neurotransmitters in this fashion to pass on the signal, right? So here in the salivary glands, right, inside your mouth, they're producing these salivary amylase and other sorts of things, saliva basically. Right? It's forming in these cells, and the vesicles that have all those particles and stuff are going to fuse and then be released, right? So one of these cells is one of these right here. And so the cells over here are making the product, and then they're releasing them into the lumen. And in this case, this is an exocrine gland. So all the product's going to be in here. It's going to be released into the lumen, and then it's going to go out the duct here and out onto, in this case, out into the mouth right there, into the external surface right there. So, you know, that's an exocrine gland, but also this is the same sort of mechanism for endocrine glands, right? So for insulin release, for instance, your pancreatic beta cells are producing insulin right here. They're budding them off. They're packaging them in these little vesicles, right? The vesicles are going to fuse with the membrane here, and then the insulin is going to be released out into the interstitial fluid and eventually the blood right here. So hormones are going to get into the blood in this way, right? exocytosis. And then the other example over here is that, I said before that these calcium channels kind of are opened up and they trigger these packages of vesicles, these neurotransmitters were already in the synaptic cleft right here. What the calcium does is kind of trigger it and cause in a complicated way these vesicles to fuse with the membrane and release the neurotransmitters into the synaptic cleft where they're going to bind to this receptor right here. Right, so this is another major example we'll see with our exocytosis, releasing packets of stuff out into the environment right there. So sometimes, in particular, when you get to the blood system, you'll see that stuff in the blood. Remember I said that things could kind of diffuse through the cell membrane. Maybe they can get through little clefts and stuff, right? Because your blood is bringing in glucose and oxygen and stuff like that. And it's going to get from the blood vessel, the capillaries, into the interstitial fluid in some ways, some stuff, right? From the lumen over here, there's particles we want to, bring to the body cells over here. So they're enter into the endothelial line, the cells that are lining the blood vessels over here. So they just kind of get brought into these cells on one side, on the luminal side, that's facing all the blood, and then just kind of gone across this simple squamous cell over here, and then released on the other side right here, right? This is called transcytosis right here. This is one of the major examples, these endothelial cells that line the blood vessels, moving it from one side to the other, right? So this is where I wanted to kind of bring up that last point right here before we break, before lab.